Chapter 44. Gastrointestinal Hormones, Introduction. Gastrointestinal, GI, hormones are the hormones secreted in GI tract. These hormones are polypeptides in nature and belong to the family of local hormones, Chapter 73. Major function of these hormones is to regulate the secretory activities and motility of the GI tract. Cells secreting the hormones. Enteroendocrine cells. Enteroendocrine cells are the hormone-secreting cells in GI tract. These are the nerve cells and glandular cells which are present in the gastric mucosa, intestinal mucosa, and the pancreatic cells. Neuroendocrine cells or APUD cells. Enteroendocrine cells which secrete hormones from amines are known as amine precursor uptake and decarboxylation cells, APUD cells, or neuroendocrine cells. For the synthesis of a GI hormone, first a precursor substance of an amine is taken up by these cells. Later, this precursor substance is decarboxylated to form the amine. From this amine, the hormone is synthesized. Because of the uptake of the amine precursor and decarboxylation of this precursor substance, these cells are called APUD cells. This type of cells is also present in other parts of the body, particularly the brain, lungs, and the endocrine glands. Enterochromaffin cells. Enteroendocrine cells which secrete serotonin are called enterochromaffin cells. Description of gastrointestinal hormones. 1. Gastrin. Gastrin is a peptide with 34 amino acid residues. It is secreted mainly by the G cells of pyloric glands of stomach. It is also secreted by Tg cells in stomach, duodenum, and jejunum. In fetus, the islets of Langerhans also secrete this hormone, Table 44.1. Gastrin is secreted from stomach during the gastric, second, phase of gastric secretion and from small intestine during the intestinal, third, phase of gastric secretion. Stimulant for secretion. Stimulants for secretion of gastrin are, presence of food in the stomach. Stimulation of local nervous plexus in stomach and small intestine. Vagovagal reflex during the gastric phase of gastric secretion, gastrin-releasing polypeptide is released at the vagal nerve ending. It causes the secretion of gastrin by stimulating the G cells or Tg cells. Actions. Gastrin stimulates gastric glands to secrete gastric juice with more pepsin and hydrochloric acid. Accelerates gastric motility. Promotes growth of gastric mucosa. Stimulates secretion of pancreatic juice, which is rich in enzymes. Stimulates islets of Langerhans in pancreas to release pancreatic hormones. 2. Secretin. Secretin is a peptide hormone with 27 amino acid residues. Historical importance of secretin is that, it was the first ever hormone discovered. It was discovered in 1902 by Bayliss and Starling. It is secreted by the S cells of duodenum, jejunum, and ileum. Secretin is first produced in an inactive form called prosecretin. It is converted into secretin by the acidity of chyme. Stimulant for secretion. Stimulant for the release and activation of prosecretin is the acid chyme entering the duodenum from stomach. Products of protein digestion also stimulate secretin secretion. Actions. Major actions. Secretin stimulates exocrine pancreatic secretion. It acts on the cells of pancreatic ductule via cyclic AMP and causes secretion of large amount of watery juice with high content of bicarbonate ion. Bicarbonate content of pancreatic juice, released by secretin, has functional significance, Chapter 39. Other actions. Secretin inhibits secretion of gastric juice. Inhibits motility of stomach. Causes constriction of pyloric sphincter. Increases the potency of action of cholecystokinin on pancreatic secretion. 3. Cholecystokinin. Cholecystokinin is made up of 39 amino acid residues. Previously it was thought that there were two separate hormones, namely pancreozymin and cholecystokinin. It was thought that pancreozymin stimulated the secretion of pancreatic juice with large amount of enzymes and the cholecystokinin stimulated the contraction of gallbladder. but now it is established that the same hormone has actions on both pancreas and gallbladder. So, it is named as cholecystokinin pancreozymin, CCKPZ, or cholecystokinin, CCK. Cholecystokinin is secreted by I cells in mucosa of duodenum and jejunum. A small quantity of the hormone is secreted in the ileum also. Stimulant for secretion. Stimulant for the release of this hormone is the presence of chyme-containing digestive products of fats and proteins, viz. fatty acids, peptides and amino acids in the upper part of small intestine. Actions. Major actions. Cholecystokinin contracts gallbladder. Stimulates exocrine pancreatic secretion. It activates the pancreatic acinar cells via the second messenger inositol triphosphate. Cholecystokinin causes secretion of pancreatic juice with large amount of enzymes. Other actions. Cholecystokinin accelerates the activity of secretin to produce alkaline pancreatic juice, with large amount of bicarbonate ions. Increases the secretion of enterokinase. Inhibits the gastric motility. Increases the motility of intestine. Augments contraction of pyloric sphincter. Plays an important role in satiety by suppressing hunger. Induces drug tolerance to opioids. For glucose-dependent insulinotropic hormone. Earlier it was called gastric inhibitory peptide, GIP. It is a peptide hormone, formed by 42 amino acid residues. It is secreted by K cells in duodenum and in jejunum. It is also secreted in antrum of stomach. Stimulant for secretion. GIP is secreted when chyme-containing glucose and fat enters the duodenum. Actions. Gastric inhibitory peptide, GIP stimulates the beta cells in the islets of Langerhans in pancreas to release insulin. It causes insulin secretion, whenever chyme with glucose enters the small intestine. Hence it is called glucose-dependent insulinotropic hormone. Inhibits the secretion of gastric juice. Inhibits gastric motility. Recent studies reveal that GIP does not show significant action on gastric secretion. 5. Vasoactive intestinal polypeptide. Vasoactive intestinal polypeptide, VIP, contains 28 amino acid residues. This polypeptide is secreted in the stomach and small intestine. A small amount of this hormone is also secreted in large intestine. Stimulant for secretion. Presence of acid chyme in the stomach and intestine causes secretion of VIP. Actions. Vasoactive intestinal polypeptide, VIP dilates splanchnic, peripheral, blood vessels. Inhibits hydrochloric acid secretion in gastric juice. Stimulates secretion of succus entericus with large amounts of electrolytes and water. Relaxes smooth muscles of intestine. Augments action of acetylcholine on salivary glands. Stimulates insulin secretion. 6. Glucagon. Glucagon has 29 amino acid residues. It is secreted mainly by alpha cells of islets of Langerhans in pancreas. It is also secreted by A cells in the stomach and L cells in the intestine. In intestine, it is secreted as preproglucagon. Stimulant for secretion. Presence of food with more fat and protein in the stomach is the stimulant for glucagon secretion in stomach and duodenum. Hypoglycemia is the stimulant for secretion of pancreatic glucagon. Action. Glucagon increases blood sugar level, Chapter 69. 7. Glycentin. Glycentin polypeptide is secreted by L cells in duodenum and jejunum and alpha cells of pancreatic islets. It is also secreted in brain. Precursor of this hormone is the preproglucagon. In intestine, the preproglucagon is converted into glycentin and glucagon-like polypeptide 2, GLP2. In pancreas, it is converted into glucagon, glucagon-like polypeptide 1, GLP1, and major proglucagon fragment. Stimulant for secretion. Glycentin is secreted when chyme with fat and protein enters the intestine. Action. Like glucagon, glycentin also increases the blood sugar level. 8. Glucagon-like polypeptide 1. Glucagon-like polypeptide 1, GLP-1, is secreted in alpha cells of pancreatic islets, see above. Structurally, it is similar to GLP-2 and glucagon. It is found in brain also. Stimulant for secretion. Presence of food with glucose in the small intestine stimulates the release of GLP-1. Actions. Glucagon-like polypeptide 1, GLP-1 stimulates the insulin secretion from beta cells of islets in pancreas, inhibits gastric motility. 9. Glucagon-like polypeptide 2, Glucagon-like polypeptide 2, GLP-2, is secreted by L cells in ileum and colon, see above. Structurally, it is similar to GLP-1 and glucagons. Like GLP-1, it is also found in brain. Stimulant for secretion. Presence of food with glucose in the small intestine stimulates the release of GLP-2 also. Action. GLP-2 is believed to suppress appetite. 10. Somatostatin. Somatostatin was first found in hypothalamus and named as growth hormone inhibiting hormone. Now it is found in D cells of stomach and upper part of small intestine. And D cells of pancreatic islets also. Somatostatin is secreted in two forms, one with 14 amino acids and the other one with 28 amino acids. Stimulant for secretion. Presence of chyme with glucose and proteins in stomach and small intestine causes release of somatostatin. Actions. Somatostatin inhibits the secretion of growth hormone, GH, and thyroid-stimulating hormone, TSH, from anterior pituitary. Inhibits gastric secretion and motility. Inhibits secretion of pancreatic juice. Inhibits secretion of GI hormones such as Gastrin. Kleecystokinin, CCK. Vasoactive intestinal polypeptide, VIP. Gastric inhibitory peptide, GIP. 11. Pancreatic polypeptide. Source of secretion. Pancreatic polypeptide is a polypeptide with 36 amino acid residues. It is secreted mainly by the PP cells of the islets of Langerhans in pancreas. It is also found in small intestine, Table 44.1. Stimulant for secretion. Pancreatic polypeptide is secreted by the presence of chyme with proteins in the small intestine. It is also secreted in conditions like hypoglycemia, fasting and exercise. Actions. Pancreatic polypeptide increases the secretion of glucagon from alpha cells of islets of Langerhans in pancreas. Decreases the secretion of pancreatic juice from exocrine part of pancreas. 12. Peptide YY. Polypeptide YY with 36 amino acid residues, is structurally related to pancreatic polypeptide and neuropeptide Y. It is secreted in L cells of ileum and colon. Stimulant for secretion. Presence of fat-containing chyme stimulates the release of peptide YY. Actions. Peptide YY inhibits gastric secretion and motility. Reduces secretion of pancreatic juice. Inhibits the intestinal motility and stops passage of bowel beyond ileum, ileal brake, suppresses appetite and food intake. 13. Neuropeptide Y. Neuropeptide Y contains 36 amino acid residues. It is structurally related to pancreatic polypeptide and peptide YY. It is secreted by enteric nerve endings, particularly in ileum and colon. It is also secreted in medulla, hypothalamus and neurons of autonomic nervous system, ANS. Stimulant for secretion. Secretion of neuropeptide Y is stimulated by fat-containing chyme. Action. Neuropeptide Y increases the blood flow in enteric blood vessels and stimulates food intake, Chapter 141. 14. Motilin. Motilin is built by 22 amino acid residues. It is secreted by MO cells, which are present in stomach and intestine. It is also believed to be secreted by enterochromaffin cells of intestine. Stimulant for secretion. Motilin is secreted when the chyme from stomach enters the duodenum. Actions. Motilin, accelerates gastric emptying, increases the mixing and propulsive movements of small intestine, increases the peristalsis in colon. 15. Substance P. Source of secretion. Substance P is a neurotransmitter with 11 amino acid residues. It is secreted at the pain nerve endings in brain and enteric nerve endings in small intestine. Stimulant for secretion. Secretion of substance P in intestine is caused by the presence of chyme. Actions. In GI tract, substance P increases the mixing and propulsive movements of small intestine, refer chapter 141 for its actions in brain. 16. Ghrelin. Ghrelin is a recently discovered hormone. This 28 amino acid polypeptide is synthesized by epithelial cells in the fundus of stomach. It is also produced in smaller amounts in hypothalamus, pituitary, kidney and placenta. Stimulant for secretion. Secretion of ghrelin increases during fasting and decreases when stomach is full. Actions. Ghrelin promotes the secretion of growth hormone, GH, by stimulating somatotropes, growth hormone synthesizing cells, in anterior pituitary. Receptors for this hormone, called growth hormone secretagogues receptor, GHSR, were identified in the somatotropes before the discovery of the hormone itself. These receptors are also found in adipose tissue, heart, and hypothalamus. Induces appetite and food intake by acting via feeding center in hypothalamus, chapter 149. Stimulates gastric emptying. Other gastrointestinal hormones. Mucosa of GI tract secretes many other hormones such as enkephalins, serotonin, enterocrine, dynorphin, neurotensin, urogastrone, villicanin, guanylin, bomazine. However, the significant biological actions of these hormones on GI tract are not clear.